Hey, Andre, does your mic work?
Hello, can you hear me? I hope so.
Yeah. All right. Just wanted to make sure that everybody is good, no technical difficulties, and that we're not echoing or anything like before. Hey, Andre, does your mic? Okay, so I think we can start now. This is the 6th day of the Ecology in R web conference series. Our last talks were about ecology in R, and we had some stat stuff previous to that. For everybody that's watching on their browser or in Zoom, we're going to have the speakers talk first, and then we'll take some questions after. This one is going to be a little bit different than the previous talks because we have 3 speakers. So we'll have a little bit more brief of a time in between to ask and answer questions. But all of these talks are going to be recorded and they'll be released both on YouTube and in the group. So if you guys want to ask any of these package authors about their packages, about the functionalities, and try to test them out, please feel free to do so. Okay, so with that said, I'll introduce our first speaker, who is Richard Shefferson from the University of Tokyo, Japan.
Okay, so let's see. I can't start my video here, but whatever. I will share my screen here.
1 second. I have to make you guys co-host. Should be good now.
Okay. Okay. So thank you very much. My name is Rich Shefferson. I'm an associate professor at the University of Tokyo. And so today I'm going to introduce an R package that was released pretty recently, about maybe officially on CRAN 3 months ago, but last month in terms of the papers about it. And let me just share my screen here. So the package is called LEFCO 3. The main point of it is to create and analyze historical matrix projection models. And so let me just kind of introduce that concept first. So I'm guessing that some of the people watching here are at least familiar enough with population ecology that they know what a matrix projection model is. And so the matrix projection model is really a mainstay of population ecology, originating with Leslie in 45 and later on with Leonard Lefkowitz in 1965, who kind of came up with the core concepts, the core matrices that are used. And this is probably the most widely used method to analyze population dynamics in wild populations. Certainly not with people, but if we're talking about any species other than humans, then this is the most commonly used method. And the typical representation you can see here, this is a Leslie matrix, which is age-based. That's the approach that he came up with. And you can see that there's a matrix here. So it's a square matrix. And it has as many rows and columns as there are ages in this case. And the S terms correspond to the survival from one age to the next. The F terms correspond to fecundity associated with each age. And the zeros are structural in this case because you can't go from, say, age 1 to age 3 in a single time step. Leonard Lefkowitz expanded on this because he noted that there were many organisms for which age doesn't explain very much in terms of demography. And for those sorts of organisms, you might have some other state variable that you can encapsulate in some sort of stage, the most commonly used being size. For many plants, size is the best measure of demography. So he came up with this approach, and here notice that there are basically no structural zeros. All kinds of transitions are possible because age is out the window now. The key problem in this, these are widely used, particularly in conservation biology, and they're very useful, but they have some problems. And from my perspective, having worked with them for maybe 25 years now, there's one really key problem. And that is that if you look kind of at a sample matrix like this one, which this is for a plant that I study, this is the small yellow lady slipper in North America, which is a very long-lived plant. And this is kind of a diagram of its very complicated life history. This is an extremely simplified version. And here's a matrix corresponding to that. You'll notice that basically what we've got are you've got an original time, and so the plant is going to be in some stage at some time, and you're trying to predict whether it will be alive and in what stage it'll be in the next time. Okay, so in other words, the state of the individual, say next year, is entirely determined in this model by what stage or state it's in now. So this assumes that there's no influence of individual history. Okay. Now, why is this a problem? So I would say that at the time that Leslie and Lefkowitz were publishing their original papers, this wasn't particularly well known. There wasn't a lot of literature about individual history and its influence in ecology. But we know now from a lot of really well done studies that events in life have long-term impacts that affect things like vital rates. So, for example, the Nussey group did a really wonderful long-term study on the red deer, Cervus elephas, and found, for example, that early life reproduction affects senescence rates. So senescence is the rate of aging, right? So if you're an early life reproducer, you start to age at an earlier time and you age more rapidly. Likewise, your early life environment affects later reproduction. So if you're born as a red deer into a really dense population, then your later reproduction is going to be lower. And that's an entire, that's due to essentially to a historical carryover effect that would not be incorporated in a matrix model. Here's another quick example. So this is early life reproduction shortening life expectancy. This is in effect a very simple trade-off, but it's completely missed in matrix models because of that one time step window. If the early life reproduction happened a few time steps back before the transition you're interested in, it won't be encapsulated in this. And in my own work, I found this to be the case. I've done a few little studies looking at really long-term impacts of very simple events. For example, this is a plant Coralorhiza odontorhiza that's non-photosynthetic. It has no reason to sprout except to reproduce. And it turns out that sprouting is so costly to it that each sprouting event has a mortality impact that carries over many years into the future, which you can see here. This is the A's are sprouting and the D's are dormant, so living below ground. And you can see that the more dormancy you have, basically, the longer you live, right? So I became really worried about this maybe, I would say, 7 or 8 years ago when I was working on a project trying to understand why it was that some photosynthetic plants didn't always sprout. This is a strange phenomenon that, some plants do this for very long periods, maybe 10 years, 20 years, when they're living entirely below ground, not sprouting, not photosynthesizing, not reproducing. And I wanted to understand it. And I started to work with matrix models and finding that the standard matrix models couldn't explain it. And all these, in the standard matrix model format, if I applied an evolutionary kind of analysis, such as adaptive dynamics, pairwise invasion analysis, it didn't make any sense. It was never adaptive. Until I found a way to incorporate a little bit of individual history. And when I've did that, when I incorporated individual history, I suddenly began picking up long-term trade-offs. For example, a growth impact. So if you're a plant that starts out small and then you grow large, in the next year, you might have a high mortality rate because of the cost of becoming large in that middle year. And so these are pairwise invasion plots for those of you who may not be familiar with this. The key here is if you see a plot like this, this is showing you a fitness landscape essentially of the tendency towards sprouting in a resident versus an invader within a population. These are competing genotypes, and these scenarios here, these are standard matrix models, deterministic and stochastic, all predict that this phenomenon shouldn't occur. But if I incorporate an extra year into the matrix model, then I have an evolutionarily stable strategy. So suddenly it makes sense not always to sprout. So this was the first warning I had about this. Now the problem is, so this is a non-standard format, and this particular kind of model where you incorporate one more year was introduced by Johan Erlen in a paper in Ecology in 2000. And he called it the historical matrix projection model. And how does this work? So this shows you an example. So if we go back to my original example of this life history that has 5 stages here, here's a matrix model for it. What we do, we need to keep the 2-dimensional matrix format. You can't like add another dimension, make it a cube, and expect linear algebra to work. It won't. So we need to keep the 2-dimensional format. And in order to do that, what we do is we, instead of using single stages on the rows and columns, we use stage pairs for consecutive times. So here we have the 2 transition. This is the stage pair corresponding to time t and time t plus 1. And on the from side, we have time t and time t minus 1. So time t minus 1 is what we're adding, okay? And so this is the key thing. And notice that the matrix gets huge. Okay, so let's take a look at this matrix in more detail. So there's a lot of zeros here. And the reason that this happens is because many of these transitions are now physically impossible because we have state at time t in both the rows and the columns. So state at time t has to be equal in order for a transition to be possible. So for example, if I have this transition, this from pair here, j and time t minus 1 and v in times t, well here we have state s in time t and state s in time t plus 1. That transition is impossible because the organism can't be in state v and s at the same time. Okay, and it turns out that these matrices very quickly, very easily become sparse. So with mostly zeros, and they can get quite huge. The projection, though, because this is a 2-dimensional matrix, is the same as before. So you can do everything with a historical matrix projection model that you can do with a normal one, including project to the next time step, project lambda, do stochastic analyses, whatever you want. Now, why should we use these? This just looks like an extra complication. I'm sure to most people are thinking, well, if I have my data, why can't I just do a standard one and look for the best? So honestly, I mean, after working on these things for as long as I have, and in particular on the conservation side, I can tell you that I would argue that most of the inferences made with matrix projection models have been pretty bad over the years. And published critiques of this suggest that, for example, Elizabeth Crone published a wonderful paper in Conservation Biology a while back showing that 40% of matrix projection-based population viability analyses project population trends accurately 5 years into the future, just 5 years. So in other words, 60% don't, which is a problem considering we use these models to actually create our management plans for endangered species. Another paper by Chowdhury Nolly very recently published suggested that only 18% of PVAs, which use matrix projection models in their inferences, are of high quality, and the rest lack typically in inference from analysis or the models that are used. So my argument is, would our inferences from matrix projection models be better if we actually incorporated that extra year? The problem, though, is that historical matrix projection models are ridiculously complicated and huge, right? And I have made them for years. They require tons of code in R, a lot of patience because it takes a long time to put them together and then to analyze them. Because they're huge, the standard analytical approaches can take forever and sometimes break down because they'll take up too much memory. And so here's an example. If we have n number of life stages, then a standard ahistorical matrix projection model will have n squared elements. But a historical one will have n to the fourth elements, right? Because now you have n squared rows and n squared columns. If you do an integral projection model, for example, where you might have 100 stages, well, now you have 10,000 rows and 10,000 columns, so 100 million elements that you're estimating in a historical projection per matrix. My first attempts to do this, sometimes it would take 30 minutes just to set up a single model. Hence, this R package, which is made to make this process simple, quick, and I want to say relatively painless. I think it's always painful to work with these models, but relatively painless. So this is an R package that's built to create raw and function-based matrix projection models, both historical and the normally historical ones. And it can even create integral projection models if you're interested in that. It's built as an R package powered via C++ based binaries, and that's where it gets its speed. So R code by itself can be ridiculously slow if you have to work with huge objects, but tossed into a binary and it'll come out quick. So that's the underlying backbone of this. And there are step-by-step guides that are being developed. Some are online now, and I actively maintain this and I put up more. And it's also on CRAN, so it's very easy to access. Here's the standard workflow. So we start off by developing a life history model. There's a function that can help you do that. That has to be very explicitly defined. And in my opinion, it's the most difficult part of this because you really have to develop a life history model that matches not only the biology of your organism, but the data set and its characteristics, what's there, which stages are actually gone through over what time periods and so forth. And then we organize the demographic data using functions here that can take most formats that people use. We also have a few functions that can summarize key data that needs to come externally from, say, extra projects that you do. And then we can test for the impacts of history as well as create vital rate models if you're interested in creating function-based matrices or integral projection models using a function called model search that actually completely automates model selection for all the vital rates that you need. Then we have a number of functions that can create the matrices themselves very quickly. FLEFCO3, FLEFCO2 would be making function-based matrices. There's raw versions as well. And I've also created an age-by-stage matrix maker called AFLEFCO2. And then I've included a number of functions for analysis that are much quicker than other functions available typically to estimate lambda, for example, or stochastic growth rate, sensitivity and elasticity analyses that are both deterministic and stochastic. And I'm working on developing life table response experiments, deterministic and stochastic, and a few other things as well. Let's see. So we're at we're at 17 minutes past. I'm going to kind of try to go through this a little bit quickly. The developing the life history model. This doesn't matter what methods you use, mine or others, but this is you have to consider what sorts of life history stages that you're interested in and whether they're actually parameterizable given your data set. So this requires knowing your data pretty well. And I'm just going to blow through this a little bit to save a little bit of time. But where LEFCO 3 comes in, I think initially as a lifesaver is in terms of organizing your data. Most people have their own preferred sort of data format in Excel or whatever they're using, whatever kind of spreadsheet protocol. And we could take that, and there are functions that will basically take most formats that you have and structure them in a way that can be analyzed in a matrix approach. Okay, so we create what's known as a vertical format to them. In this case, it's historical, so you'll have 3 states for each individual across time, along with individual ID tags, so you could run mixed models with them and so forth. This is an example here of a horizontal formatted format data where you have each individual takes up 1 row and all of its states across time, for example. So we can work with that here. Let's skip ahead a little bit. Modeling the vital rates. So here we can test whether individual history is important on any vital rate, as well as create the vital rates and the vital rate models for the function-based NPMs and integral projection models. So this is done via one function right now called model search. This actually estimates survival, observation probability if you're working with an organism that is not necessarily always seen. So if there's like an unobservable life stage, you can factor that in. Size transition probability, reproduction probability, and fecundity rate. These are all conditional probabilities. So this will take care of subsetting the data properly for you to estimate them all. And then you can choose to run mixed models versus GLMs. If you run mixed model, then you have to have individual identities in there so that you can cut down on pseudo-replication in there. We currently handle binomial Gaussian Poisson, negative binomial, zero inflated versions of Poisson, and negative binomial distributions for all of these. So you have a lot of flexibility. And I'm going to be adding more flexibility with particularly GAMs. That's one thing I'm going to add at some point. The factors that can be tested, size. This is really focused on size classified stages. So size is a part of this. You can test size, reproductive status, fecundity as factors determining the vital rates. Individual and environmental covariates of various kinds, age, patch identity, the actual specific time, and individual identity. And particularly for size and reproductive status, we have historical and ahistorical states for those that can be incorporated. And then modeling is automated. It'll develop the global models for you depending on what you want. It'll then dredge essentially, so it'll use the dredge function in Moomin to come up with all the different models that you want, and it'll determine the best fit. It can actually determine the best fit as the most parsimonious, so not necessarily the lowest AIC, but something, it could be something closer to it that has fewer parameters, and so it can determine that as well. The matrices themselves that it can estimate are going to be historical and ahistorical. It can handle both. It can handle raw versus function-based and integral projection models, fully stage-based or age by stage. I don't do any pure age models in this. And so these are the functions that could be used for that. And let's see, and then we can analyze matrix projection models. And so this is where I think we get a lot more use out of this, because having worked with, say, the Pop Bio package and what's the other one? There's another one, Pop Demo. Those are really great packages, but they assume you're working with dense matrices. And so if you fit in, say, a matrix that has 100 million elements in them, you will blow up your computer. So these methods are all built to handle those extremely large matrices in quick time and without overloading your memory. And so these are the current functions that we have for analysis. We can create element-wise means, lambda, stochastic lambda. You can calculate the stable stage distribution, reproductive values, sensitivity, elasticity, deterministic and stochastic in both cases. And we also have a few other things that are kind of useful. There's a few different kinds of summary statements that can really collapse those huge things down into something meaningful for you, like elasticities according to transition rate and so forth. I want to do a quick demonstration. I guess we're 22 minutes in, so we still have some minutes. I thought I would just do a really quick demonstration in R of how this might work. It's very quick. Hopefully that'll show you kind of what this is about. Let me move over to R very quickly.
There's no set time limit, so
there's no set time limit? Oh, okay.
No, go ahead.
Well, I'm not going to take you up for 2 hours, although I could. So I thought I'd move this over here to RStudio. This is just a really quick tutorial that I put together. And what I'm going to do, I'm going to run just the whole thing and have it work on that, because there's one step in here that takes maybe a minute or two. But what I'm doing here, there are two data sets that are included in the R package itself. Both of them are plant-based because, you know, plants are where we see size classification being really, really important. But the key, you know, so here I load in one of these data sets. This is the data set that Johan Erlen originally published 21 years ago when he introduced this kind of model. And so this shows you what the data set is structured like. It's just a quick summary of it. Johan likes to, you know, take his work and put it into this kind of horizontal format where each row is all the data for one individual. And that's great, but you need to be able to collapse that into a vertical format in order to create these linear models that can encapsulate your vital rates. So later on, there's a step where we'll do that. Here we create the life history model. There's a few steps here. Basically, these are vectors that correspond to the different sizes that we want, as well as the names of the stages. So here are the names of the stages that I'm going to be using. Here are the centroid of the size range. You know, there are always size bins involved in this. There are, whether these are reproductive stages or not. So this is a binomial variable. This is whether or not you can observe it. This is whether it's a mature stage, whether it's an immature stage, whether it's a propagule stage, and whether the stage occurs in the data set. So this package needs to know that in order to create the vital rate models and to populate the matrices. And then this is just giving you a half width of the associated bin. And then sfcreate here creates this data frame that the package is going to use to create the other structures. And we have a comments field as well that you can, you know, tell yourself what on earth you've got. Hold on, RStudio sometimes does things you don't like. Anyway, so for example, this is SD corresponds to dormant seed stage, SDL is the seedling stage. So just to remind yourself, okay. And so this is what the data frame looks like. You see, so you know everything is put together in this order that can be used essentially both by R and by U to remind yourself what everything is like. The next step that happens after that is a reorganization of the data set. So there's this function here, verticalize 3, that'll take a horizontal data set and turn it into what we need. And what we need is for all the data for an individual to be broken down into sets of 3 consecutive times. So what is its state in times 1, 2, and 3, and then times 2, 3, and 4, and times 3, 4, and 5, and so forth. For the standard matrix model, that would be all you need, but I think there's enough criticism of that approach and of the fact that it's kind of pseudo-replicated that we need a little bit more typically. So this also keeps track of the individual identity, which becomes a factor in a mixed model, if you're interested in that approach. So all that is incorporated here, as well as a few other things, the observed age, when the organism or the individual is first seen and last seen, and so forth. Its stage, so this automates the actual stage classification and so forth. The next step is just to provide a little bit of extra data that we don't have in the data set. So in this case, we don't have germination rates and we don't have seed dormancy rates. So we provide them extra in here, as well as what are really the reproductive stages and what do they lead to the reproduction of and at what rates. So these are multipliers on that. We incorporate. We build vital rate models here. So this is the function that takes the longest. There's really no way I can create a C++ version of this, unfortunately. So it'll create the global models, it'll dredge the models and so forth. This can take up a lot of time if you build a particularly complicated model and have a very large data set. But in this case, this data set is small enough that it only took about a minute or two. There's a lot of output that it gives you just so you can keep track of any weird things. If you're used to, say, working with packages like LME4 or NLME, you know that all this red is normal, so this is not worrying. It would be worrying if it stopped, and it didn't. So it finished with that. It developed the best fit models. We can actually summarize what this looked like. I'll just very quickly show you that. So if I do a summary, here. I'm sorry, summary is not a function, summary is, right. So this is what the summary looks like, and it tells you quite a bit. So the summary will include the chosen best fit model according to which metric you're using to determine that. In each case, so we've got a binomial model here, these are probabilities. The size model in this case is a Gaussian model. It's an LME4 Elmer object. So this tells you what's in that. Reproductive status is binary or binomial. And then we have a Poisson distribution for fecundity that you can see here. And this is the best fit model here. History shows up as being incorporated here in the size model. This is size in time 1, which is required to know size in time t+1. So we see that individual history is important. Let me see. So going back down here, the next step here creates the matrices. And so I just summarize them here because this is creating historical matrices. They're rather huge. The summary of that tells you that it created 18 matrices. Each matrix is 441 rows and columns. This is the total number of elements. It tells you how many transitions were actually estimated because most of those elements in the matrices are zeros, structural zeros. And it tells you how many individuals as well as individual transitions were used to propagate the vital rate models that were used for the matrices as well. This is actually one of the matrices, so you can take a look at it itself. It's, as you can see, it's highly sparse. It's mostly zeros. You can go, it'll only show you about 50 columns at a time, I think. But you can go and zoom in on estimated stretches, for example. And once you've got that, you can analyze it. So here we see lambda. So the lambda estimator here will calculate lambda for all subpopulations as well as overall for the population, as well as for the annual terms and so forth. So this is showing you lambda from 88 to 89 for this population, this patch going forward. And down the line, here we can see the equivalent mean. So the element-wise means. This is showing you the overall mean lambda corresponding to these patches, and then this is the overall mean for the population as a whole. If you want stochastic lambda, I ran that very quickly. You can change this, but this assumes that you want a stochastic window of about 10,000 time steps. And what you can see here, this sort of matches up at least qualitatively that you've got a lambda below 1 here, you've got a negative log stochastic growth rate here, so the population is declining at the patch level and overall. We have the elasticity analysis here. And the nice thing about this elasticity approach, so you can do both deterministic and stochastic, and it'll spit out both. If you're running a historical matrix, it'll spit out the overall historical elasticity matrix, but it'll also do a collapsed version that'll tell you just the normal ahistorical elasticities associated with that. So this is the ahistorical version, which is 21 by 21, much easier to look at. And it'll summarize it for you too. So using the summary statement here for that object, and it'll tell you, for example, so for each of these matrices, what are the summed elasticity values for, say, stasis transitions, growth transitions, shrinkage, fecundity, so forth. Notice that fecundity was completely ridiculous, just it doesn't do anything in these matrices because this is a very long-lived plant. There's very little variation in fecundity overall, so it's interesting that that just bottomed out. This is the ahistorical elasticity. It does the same for historical transitions, which are a bit more complicated. Like you might have growth transitioning to stasis, stasis transitioning to shrinkage, and so forth. Okay. All right, so that's, let me see, that's just the run through there. And the current version that's on CRAN is 3.3.2, and that's basically everything that you saw there is what can be done. The next step that I'm going to do is to add life table response experiments, deterministic and stochastic. And in the further future, I'm going to be adding density dependent projections, GAMs, and invasion analysis as a part of that, as well as adding more vignettes and instructional videos, which are currently available. So you can access everything. You can easily install it in R. If you just type install packages leftgo 3, it's on CRAN. So to do that, there's a paper introducing this that you can take a look at, and you can see my web page or just the CRAN site to see what the latest is on that. Now with that, I'd like to thank you.
All right. Well, that looks like a really, really robust package. I saw in your first slide, I think it was, that it looked like you had a visualization of what seemed to be maybe a PCA plot with like a overplot density. There was like kernel densities on there. I expect that the returns on the models are returning that large S4 object. Is there plotting functions in this package, or is it just...
right. I did not yet create any plotting functions in here. Right now I'm working, I'm instead using plotting functions elsewhere for small dense matrices. There's a PopBio has one called image2 that's kind of useful, but it blows apart when you have a large matrix. So there's also, I think the sparse M package, there's that package is all about converting large matrices into sparse format and then visualizing them. So far I'm using that. Creating good visualizations is definitely on the drawing board, but I'm still kind of thinking about how best to do it, and I haven't really decided.
Yeah, that makes sense. Usually on some of the matrix or population model, the other packages, that they default to base R plots with their resulting S4 object. But then a lot of the time in the group, I'll get questions on how do I accurately or effectively extract the data from that export object so I can plot in ggplot. I can do all these other stuff. So I'm sure that's going to be the first couple of questions that you're going to get.
Well, the one of the core things I really wanted to do here, I wanted basically just about anybody to be able to use this. And so that basically required me to do this in so the objects that I develop here are all S3. They're all basically lists, and they're going to be lists of matrices. Input and output, the inputs are going to be vectors and data frames for the most part. The outputs are basically lists of matrices or lists of matrices. So they're easily accessible because of that, right? In an S4 object, you're going to have issues probably because of the complexity of S4 objects, right? So I've tried to work with that as much as I can so far, but I haven't really figured out the best approach for plotting yet. And yeah, if you have suggestions about that, that'd be great.
All right. I also have one more question regarding the documentation that you have on the rdrr.io.
Oh, yeah.
It says under getting started, you have 4 bits of documentation. One is, for example, basic theory and concepts, and there's 3 more. If somebody was just getting started with matrix population models, would you suggest any of those or all of them?
Yeah, I would. I definitely do that. I do warn you where I think you need more, right? Because, So the four that are listed up there, I actually wanted to include a lot more, but I don't know if any of you have interacted with CRAN at a developer level, but CRAN has very strict rules. And it turns out that there are strict limits as to how much PDF you can put on there. So I put basically as much as I could. And I've been developing extra things as well. What the key thing, though, that I think particularly grad students who get into population ecology don't realize is just how important it is to understand the underlying sort of ecological theory behind the life history model itself. That's what I think people need to focus on, because a lot of the stuff that gets published, actually you wind up having analytical errors that are caused by improperly parameterized life history models. For example, somebody will develop a pre-breeding model and then include an extra transition that doesn't exist because a pre-breeding model can't have something like that, right? So for example, some people put in survival over the survival of newborns in twice by accident, by adding a newborn life stage to something that shouldn't actually have a newborn life stage because it wasn't defined that way, the way that they were actually doing their monitoring. That's the stuff that I don't get into there, but I warn people about that. But everything else, I provide at least enough of a sketch that you know what you need to study. Hopefully it's enough for that. Yeah, I do kind of in the long term want to put more long format documentation on my own website that'll be available for that. But for the package itself, which is what you're seeing there on the ERDRR site, that's about as much as I could put in without pissing off the CRAN folks.
Yeah, and good point about understanding the underlying concepts of it. I think you really highlighted that with saying or quoting that study that says about 18%, I think you said, were actually effectively carried out. So I'm still waiting for questions. If anybody on Zoom or anybody on the YouTube feed wants to ask, I'm open to both and I'm monitoring both right now. In the meantime, I'll just keep asking questions because I have a lot. On one of your slides about model vital rates, I think it was, you said there was something that corrects for unobservable life stages. What would be an example of that?
Yeah, so I actually got into this by working on an organism that had an unobservable life stage, which is vegetative dormancy in these herbaceous plants that live a long time. Sometimes they don't sprout, so a lot of orchids, for example. I studied orchids in the middle of the US, and they're often endangered. And so because they're endangered, management agencies really want to understand their population dynamics, but as they generally track their, what individuals do over time within a population, but a lot of times they'll wink out. The thinking is that they're dead, right? Because, here you see this large plant one year, the next year you don't see anything. 3 years later, there's something in the same spot. It must be a new plant. Well, a lot of these plants are actually capable of living underground for years at a stretch, okay? And if they're capable of doing that and you don't see them, then from the point of view of the demographic analysis, it's almost like you're missing data, right? So the way that you can deal with that is by creating a life history stage that's completely unobservable. If it's unobservable, but you can infer most of the instances of where it occurs within your data set because of, say, the position of the plant, right? Because maybe it's not a very dense site, if it occurs right in this particular spot, right by some flag or something, it must be that plant, then you can tell it's in that stage. You still have to make a few rules or do some extra analyses to figure out what the likelihood is that it's still alive. But if you can do that, then you get a new vital rate out of that, which I generally refer to that as the observation probability. But for a plant like this, it would be the sprouting probability. Okay, so the chance that it survives, assuming it survives, does it sprout? If it does sprout, how big is it going to be? That's the size transition part. If it does sprout, then is it going to flower? That's all on top of that.
Okay, well, that definitely answers my question. I'm gonna give it another little while to see if anybody else has any questions. I think you explained it pretty well in your talk, so.
Well, I hope so, but you know,
Yeah,
I'm happy to take questions by email too, or whatever.
Again, anyone using Zoom right now, I'm looking at the Q&A dialog box. So you can either post there, or anyone that's watching the live stream on YouTube, please feel free to ask any questions, comment on the package. If you're working with matrix population models, maybe you have some questions about your own research, now would be a good time to ask. Oh. Okay, well, again, we're going to have these recorded and posted to the page. So please feel free to reach out to Mr. Richard Shefferson, and you can check out his GitHub page, the RDRR link that I posted, also on the YouTube feed if you want to look over those documents that I was talking about earlier. And thank you very much for giving this talk right now. I'm going to hand it over to our next speaker, and we can also continue the discussion on the Facebook Ecology and Art group whenever I post this link. So thank you again. And our next speaker is going to be Mr. Ondrej Mottl. Please forgive me if I mispronounce that, from the University of Bergen, Norway.
Hello. Okay, that's all. My name is Ondrej Mottl. I think mispronouncing my name is pretty common, so don't worry about it. It's super. Now it's super okay. It's super hard. We are the only language which has r in there, so it's normally no one can pronounce this. So I'll try to share my screen. Yeah, I hope everyone can see this. So I'm super glad. I will thank Professor Shefferson for the talk, and we will continue with the topic of age and time, but I think we will start talking about rate of change, therefore, you know, vegetation change per time, with a special focus on paleoecology. So it'll be a little bit change of a topic, but I think it could be very interesting. So this will be overview of my presentation. I will try to do it in about 30 minutes. I will start talking a little bit something about myself, so you'll understand how I get here and why I'm doing this. A little bit, I would do a little bit background to paleoecology, what's the differences from normal ecology, which I think will be important to understanding why the package is important, and why is it so cool, and why is it doing new, I would say. Then I would show how the package is working and how to use it, and then in the end I will show some technical usage of the package in the R. So as I said, my name is Ondrej Mottl. These are some ways you can contact me on the email or on the GitHub. I'm from Czech Republic. I studied at the university. My PhD I studied at the University of České Budějovice, and now I'm working at the University of Bergen in Norway. So I consider myself an ecologist, and I was working on tropical ants in Papua New Guinea on community ecology. But through my study of my PhD, I developed really passion and interest in statistics and in, I would say, data science. And therefore, I little bit switch a field, and I jumped into something which is called more interested into like data-heavy computational thing, and I jumped into paleo. And now I'm in a project which is called Human on Planet Earth, or HOPE. And we are basically trying to answer one simple question, which is, did prehistoric humans alter the fundamental ecological processes in the last 10,000 years, right? And this is an overview of what we're trying to do. We are basically taking a pollen databases and archaeological surveys, archaeological data, and we are developing new numerical and statistical tools to try to look into the past and see if the humans in the last 10,000 years affect these ecological properties. So this is a little bit overview of the team, what we are doing. It's led by Professor Bergs, both John and Hillerberg. There's also Professor Seddon, Professor Greitz. There's also this amazing team of postdoc, which I would like to do shout out to. They're great to work with. And if you want to know more information, there is a webpage. You can go and check it out. Now, a little bit to introduction to the paleoecology and time series and pollen analysis in general, which I think is important for those who don't know how does it work. So you will know how does it work, so you will understand the package. So a little bit broad background is that we know that the world is changing, right? We know in the last 10,000 years, since the last ice age, there was this huge warming up since the last ice age, and now there's even another warming up due to human activities, right? We also know that the humans change the way they are living on this planet, and there's been this massive shift in the way we are doing the agriculture, and there's this massive shift to the very intensive agriculture on the world on the global scale. We also know that environment is changing, not environment, but like biodiversity is changing, and we can observe these changing. And this is a very interesting study, which was published in Science 2 years ago, where they took 50,000 different basically time series of biodiversity and see how quickly different biodiversity changed, right, in time. So this is turnover, basically species turnover in time on the land and on the marine. And you can see in the blue is slower changes, in red is faster changes, but in general we can observe that the world is changing. If we want to look how quickly or slow or how the changes are, we want to use what is called palaeological sequences. And they're really cool because they're a source of information where you can use it to look beyond this time of human observation right now, beyond of possible experimentation where we can observe right now and look to the past. So what are they? What are paleological sequences? We call them a window to the past and how they work. The simple principle how they work is basically like this. You can imagine there are some trees and there are some fauna composition which they produce pollen, and they fly around in the air. Or you forget about the charcoal right now, it's a little bit more complicated, but imagine the pollen particles are flying away, and there's a lake somewhere, and because they're a little bit heavy, in the time they will slowly fall down onto the lake, and they will be on top of the lake, and because they're again a little bit heavy particles, during the time they will slowly sediment down and lay on the bottom of the lake. And if you wait, and they will make a layer, and new pollen in 1,000 years will again fly in the air and sediment down and create another layer. And if you do this repeatedly through time, through 1,000 years, what you'll end up with is a layer after layer of sediments, and each layer including basically composition of pollen from plant composition around the lake in the time. And what you can do, you can dig a hole and extract what we call a core, which is basically, like several meters long dig, where you can see each of these layers is individual small window to the past, which has particles of pollen from that time. So what paleoecologists can do, they will take samples from this core. You can imagine this being a core, which is some length, and the depth. And at several parts, you will take samples. We call them levels. And from each of these samples, you will look under the microscope, and this is what you will see. These all small parts are pollen particles, and some very amazing people can actually identify them to a certain taxonomic level. And you can say, this is Pinus, or this is some other pollen taxa. And therefore, you can do this for all the samples. And what you will end up with, this is something what ecologists know, is a matrix of samples by taxons and their abundances of how many pollen grains you will find in each of these samples. So this is basically species composition matrix, right? Because it's samples by taxa and their abundances. But paleoecologists likes to do it a little bit differently, because you can imagine that certain levels or samples have certain amount of pollen grains in them. And this pollen grains is not constant, right? And it can change. So you can see this is total number of pollen grains in each of these levels. And this is you can imagine this is the species composition. You know, each of the colors is different species. So what they like to do is they like to calculate a proportional concentration or proportional abundances of the pollen in each of these samples, right? And you will end up with something like this. This will be each of this column is a taxon, and the curve is the proportional percentage of the pollen for each of the taxa in time. And you can see what's the changes of their basically proportional abundances of the taxon through the time, not through time, but through depth. Yeah. But as I said, it's the depth is cool to have, but it's much more interesting to have an age, right? Because the depth is not telling you much. And you want to actually compare two different, for example, cores to each other, then you will need to have actually time to tell something interesting ecologically, right? So how to get a time? You need to get radiocarbon dating. So what you'll do in addition to all of your samples, which are these blue circles, you will also take another, mostly less number of radiocarbon samples, which are these orange squares, put them to this machine, which will radiocarbon date them. And this will tell you, and you will construct what is called age depth model. So these are these orange squares, which you know the depth where they were, and this amazing machine, the radiocarbon machine, will tell you how aged they are, right? What's the age of this sample, and you will construct what is called age depth model. This is a probabilistic, model which tells you each of the depths, what's the probable age of that, and you use that to project to all the other samples what are their ages. But because this is probabilistic model, and as you can see, in some selections there is quite a lot of uncertainty of what is that age for a certain depth, right? We can imagine for this 400, it can be something between I don't know, what is it, 7,500 years to 5,500 years, right? That's a kind of big space. So what do you have to do? You will gather for each of these, you will gather sequences of maybe possible ages for each of these samples. So you can imagine one line being a sequence where there are always the levels or samples in a sequence, so there is never a time swap. But they can be different because you have different probability of this sequences like this. So you will gather, I don't know, like 1,000 of them or 10,000 of them. And then you will calculate a median value of those for each of these samples or levels, which will be the age of your sample. And you will end up with the age of the sample and their compositional number of pollen grains. And you will end up with something like this, which is a typical, which is called pollen diagram, which is showing you the age of all the samples and their relative abundance of the pollen for, let's say, I don't know, Alnus, Petula, Pinus, etc. And we can see this is, for example, from nowadays to 8,000 years ago, right? This is calibrated years before present. This is important. And this is how paleoecologists like to show age. This is weird, from 0 down. Ecologists like to switch them 90 degrees, right, in the time scale, but paleoecologists like to do it like this. Okay, so this was a little bit background information about how paleo works. And now to the rate of change, right? So rate of change is a magnitude of compositional change per unit of time, right? So what we want to do, we want to gather this and produce something like this. So we want to gather this multivariate information about the species and produce one single line which will tell us what was the rate of change of this community or assemblage in this particular location in time, right? And this will gather us, okay, show us how quickly or slowly it was the community changing, right? This is rate of change score in time. But there are some issues. We know that there is uneven time distribution of time intervals between the stratigraphical sequence. So, and you can see it here, we have some density of the samples, right? So this is the density of the samples, and we can see they will never be even. And this uneven distribution of the samples can cause us theoretically an issue, because if we are calculating the rate of change between subsequent samples, if we have more samples in certain location, there's higher probability that we'll just detect high rate change just by chance. So that's an issue. We also know that the taxonomic richness of the whole community, of the whole call or site, can influence the rate of change, which was shown in several papers previously. And also there has been a lack of standardization between the studies. And therefore, there is no way to compare this rate of change between multiple paleoecological studies. So hopefully this is where my package comes in and solve these issues. So my package is called R-Ratepol. It's hopefully easy to use package. And it has a novel method to evaluate the rate of change, which has including the taxa standardization and including the age uncertainties from the age-depth models, as I showed previously. It's also a very modular tool, so it can be used for many different data types, including pollen or other different paleo data types, but not only paleo. It can be theoretically used for any other ecological time series. And we also developed included the methods for detection of high periods of rate of change, which we call peak points, which are, I think, very interesting, and I will speak more about them later in the presentation. So this is a little bit overview of the workflow of how the package is working, and I will go through all of them steps individually. So how does it work? Before we jump into the individual steps, there is intersection about something which we are calling working units. Because what you can imagine, the rate of change is always calculated between subsequent working units or steps, right? You can imagine being between two samples or two levels, but it can be calculated between other different things. And we know that, for example, that some of the samples can be unevenly distributed in time. This can be problematic. So there are ways how to fix this. If you don't want to calculate it between each individual levels, you can do interpolation of time. So you can interpolate a level samples by same age differences, but then you will lose some of the information, and this is unprecise, right? You can also bin samples together by some time interval, where you will say, okay, let's give us 1,000 years and just pack everything together. You can do also selective binning, which is basically you will do, let's say, 1,000 or 500 years interval, but inside of the time bin, you will just select, let's say, one sample, which will be randomly representing that time bin for the time. Or you can do binning with the moving window, which is a method we kind of developed, and I will be talking a little bit more about it in the future. But for now on, I think it's just worth remember that working unit, this is something you are calculating the rate of change between, right? It could be levels, it could be time, it could be various things. So now, step by step, how the package is working. First step, of course, is data preparation. So you will take your assemblage of your data, which is, it can be abundances of the pollen, it could be abundances of any other thing. You will take also your information about the ages of each of your sample, and you put them together. This is automatically done in the package in some form, which is as free structure actually inside of the package. And during this step, you can do also something which is very common in Paleo, which is smoothing of the data. So if you remember, these are the very wiggly curves for the pollen in time. And something that paleoecologists do quite often is they will take this one of the wiggly curve of the pollen. This is, for example, one of the taxa. And then we'll use some of the smoother to smooth it in time to get rid of, this sampling imperfections. And it's already built in the package, and you can choose to do it or not. Then another step is selection of one-time series from the age uncertainties. If you remember from the age-depth models, you will gather these 1,000 basically sequences of ages for these individual samples. And what will happen is we will just pick one, which will be for that run or for that time selected as this is the time for the samples for now, right? And then we will get to the binning of the moving window. This is a way how to get rid of the problem of the levels being unevenly distributed in time. So what will happen? You will create the time bins, which is pretty easy to, let's say 500 years, right? This is 500 years, let's say. And then you will shift it a little bit, but still keep the 500 years, and then shift it again. And then you do it again and again and again. And you see, if you look at this diagonal, or I don't know, then you will basically can imagine this being very slowly moving the window through the time and creating the selection of moving window through the whole thing. So you can create, if you know what's the size of the bin, and then how much you want to move the window, you can theoretically pre-create all these bins already before, right? So then if you know all the bins and you know the time of each of these sample, you will just subset all the levels inside of your time bins as it's shown in this figure, hopefully. This is a time bin, and in it there's lots of samples, and one will be selected as a representation of the bin. And in another bin, another sample will be selected, etc., etc., and it will be done for all of the bins. Then another possibility which you can do to get rid of the problem of the high richness influencing the rate of change is you will take your counts of your pollen counts, your abundances count, and you can do standardization, right? Imagine if you're talking about, again, pollen analysis, you can do, if you know the pollen counts of your total samples or levels, then you can say, okay, give me some threshold value, which will be 150 or the lowest amount of what you can find in your data set, and it will automatically subset a certain amount of that abundance without replication. Therefore, you will end up in each working unit with the same amount of abundance, of same amount of individuals, right? And then you will actually get to the calculation of the rate of change, which is basically, it will take two working units, levels or bins, and it will calculate what's the dissimilarity coefficients in the multivariate analysis between these two. So from all the taxon between these two was the dissimilarity coefficients, and you will divide that per the time between these two working units, right? And this will give you one value of rate of change. Then you will do another one, the next two, subsequent one. Again, this is the dissimilarity coefficient of the multivariate analysis divided by the time between these two. This will give you another value of rate of change. And you will do it between all of them in all the time bins. And then you will just summary all these together. And this will give you the curve of what's the rate of change in time per your whole site or core. This is how we call them. But of course, because there is this random effect of A, you're randomly selecting one H sequence from your H uncertainties from H models, and B, you are randomly standardizing for your abundances, which all of these are optional. But if you're doing this, you can do also multiple randomization, right? So you can imagine this whole sequence as a single run. So selection of the H's, then subsetting of the levels, standardization of the abundances, calculation rate of change, and a summary of those as a single run. And you can do many runs, right? Let's say 10,000. So the random effect is happening here and here. And if you do 10,000 of those, then you can do summary of those. So you will gather 10,000 of different rate of change curves. And you can put them together and create like a median value, which is the orange curve here. And basically the error wrapper, which is the 95 quantile here, which show you the error bars around that, your calculation of your rate of change. And the last step, what is actually happening is that the most interesting part of this rate of change curve are actually these very sudden and high increases of the rate of change, because you can imagine in the time you are very interested in when sudden something suddenly increased, right? It can be sudden climate change, where suddenly the species composition change to that, or it can be human influence, where suddenly humans came and cut lots of forest, right? So these are actually the very interesting parts in the site or in the composition. So you're interested in these rapid changes and compositional change, right? And therefore, we included this detection of these significant peak points, what we call the peak points. And there are various methods how to do this. And these peak points are very interesting because you can then use those and compare those among studies. And I will talk more about it a little bit later also. So a little bit of summary of the whole workflow of how the calculation is happening. So you extract your data, where you can also smooth the taxa if you want. You will create your time bins. You will do a single run, which is selected of the age from the age uncertainties, subset the levels in each bin, standardize the abundances, calculate the rate of change, summary of single run, repeat this run, let's say 10,000 years, summary of all your runs, and then the peak points. You will end up with something like this, hopefully. As I said, hopefully it's a module tool, so it can do quite a lot of things. So just with the step of data smoothing, you can select now, I think, 5 different smoothing currently included. You can select just none. No, I don't want to smooth my data, but there's also other very common used smoothing types of nowadays in paleoecology. Also, there is 5 different dissimilarity coefficients used actually in the calculation of the rate of change, based on what kind of data you want to use. There's the core or chi-square, which are very used for Poland. There's also Gavr distance or Euclidean distance, which are used for other types of data. It's up to you to select which is appropriate based on what your data you are using. And we currently use also many different types of peak point detection based what you are interested in. And much more. You can see, do you want to include the age uncertainties? Do you have these age uncertainties from age evolus? If not, you don't have to include them. Do you want to standardize your data by the abundances? Yes or no? If yes, how, etc. There are many settings of how you want to do this. Yeah. So why to use this package? I think could be a good question. So we shown in a paper, which I'll show later, on artificial data, where we simulated data with a known point of change, where we know the change was done, and then we used different settings and to test where we can detect actually which of the settings will be best at detecting the place of the change. And we show that if you use individual levels or bins or the moving windows, that the moving windows, our methods, was the best at successfully detecting these places of the sudden change in the known area with relatively low amount of false positives. And also, I think the really big advantages nowadays is that instead of comparing the magnitude of the rate of change, which can vary even though with different aspects of different sites and cores, you can still use this position of these peak points of these sudden increase of rate of change. As we see here, these are 2 different sites or cores. And if you're interested in, say, synchronicity of some events, it could be climate or it could be human effects or impact, right? In a certain region, we can use this and see if we detect synchronous changes there, and we see, no, there's actually 2,000 years difference between these two, even though they are very close related geographically. So there must be, there must be something else happening in these cores, right? As I said, all this is in a paper which is currently in a review, but you can already see it as a preprint, which is available here, if you're interested. And I will show some practical examples. Also, I will not start R, but I have the code here, but I think it's okay. So currently, sadly, it's not yet on CRAN, so it's just on the GitHub. So you have to use Git DevTools to access it on GitHub. You just attach it with the library, and then I already included some example data in it. So just access the example data. That's very simple. And this is just, again, reminder, this figure I showed from previously, it's there. This is the example data. it should look exactly the same if you plot it. And the package is just three simple functions for you. That's why I thought it's easy to use. And the main function is actually called estimate rate of change. And the argument's very simple. It's data source community, which is basically your community matrix, where you put your as you would put in a vegan and any other where there is a species by samples. There's also the data source age, where you put a similar matrix, but instead of species taxa, you will put their ages for your samples. And this automatically checking, you have to actually name one column as age, but it will tell you if you don't. And for you, then you can also add age uncertainty matrix if you have them. If you don't have to. Then you can select some of the smoothing method if you want. I select age weighting smoothing. You can select the dissimilarity coefficient. Since I'm using pollen data, I'm using core distance because it should be the best. And here, this is the working unit selection. This is if you want to calculate it between individual levels or between bins, etc. So I'm selecting the moving window, which because I show it should be the best approach. If I want to standardize the data, this is the taxonomic standardization. I say, yeah, true. How many randomization do I want to run? I say, let's say 10,000. And it's currently using also it can be run on multiple threads using parallel. So using thread, it will automatically detect how many cores you have, and it will run it. So it will take a minute or two, depending how much your computer is fast. And that's the first thing. The second thing you can do is just to detect peak points if you want to, which is a separate function for now. It will not do it automatically, because there's various ways how to even detect. There are methods of how to do this. And you can read more about it in the README file on the GitHub. And the last function which is there is plot rate of change sequence, which is just basically a wrapper using ggplot. And it will just put the sequence there, and it will should show you something like this, which is the rate of change curve again, with the error bars in time, with the points where the peak points are. If you want to know more, this is a link to GitHub, or I put bit.ly short link, so you don't have to write it all if you don't have the presentation. But I put the presentation in the Facebook group, and I will try to put it on the YouTube if I'm able. And it's all from me. I hope it was okay. And please feel free to contact me with any information about the package on my email or on GitHub or anywhere else.
Great. Thank you for your presentation. Again, I'll take questions from both here from the attendees on Zoom and also on YouTube. So if you have any questions, and please feel free to ask and I'll read them out. I had a couple questions from some of your parameter slides. You mentioned on the grain count slide that you can correct for abundance via like bins or different methods, right? I think, or wait, was it smoothing? It was something that seemed similar to the hill number. You said you were standardizing on the abundances. Is that like the hill number if you're working with community ecology?
Yeah, not yet. So this is something we already been discussing quite heavily, and we have some previous already comments on this. It's currently not. So this is about the standardization of the richness of the taxa of the community, right? So we do currently what we do is something which is very common again, and this is very maybe specific for the paleo, is that you will have your total number of pollen in one of the sample, and you will just randomly subsample to certain amount of pollen grains to and it will just abundances, which is very simple. But as been pointed out already, and we've been thinking, what you can do, you can just do very similar thing where you can say subsample to certain hill number, right, or something like that. So you can do much more intrigue way of subsetting actually the community to a certain richness, right? This is something we've been heavily discussing, and it's probably going to be introduced in the future in the package, but right now it's not.
Okay. So yeah, so you could have more control over how you standardize it in that case then. That makes sense.
Yeah, exactly.
The other question that I had is, when you were mentioning binning, you went over 3 examples of times that you could bin over. What would be the most effective method of selecting time bins? Just going through them and analyzing visually, or is there a best way to do it?
Do you mean time bin in terms of a size of a time bin?
Yeah.
Yeah, so the time bin selection is a little bit tricky, especially in the paleo, because there's multiple factors going in. For example, there is the density of your samples you have, how far they are from each other. There's also the age uncertainty of the ages of these samples, right? Because we know from the age depth models this will be unprecise always from the paleo, and also there's this limitation of how precise they can be, and we can. So nowadays, I think mostly it says if you're working, it's a time scope of 10,000 years, which is last Holocene, which is where most paleoecologists are working. The very common ground is do 500 years, because this is generally okay for still see some reasonable resolution, but not going crazy in terms of what you can push your age depth models to, because there always will be unprecise. If you want to go more precise, then it's going to be very tricky, especially in, let's say, the more go deeper in time you go. Let's say, if you go hitting 8,000 years, 9,000 years, then if you go small time width, let's say 100 years, then it will not tell you much, because actually the time errors in everything start to be so big that the information is just going to be not so good. But if you're just focusing, let's say last 2,000 years, then of course you can shorten your time bins to make it much smaller, let's say 100 years or 50 years, and you will still get reasonable information. So I think it's up to every scientist to actually go into their data, look what they have, but also be always aware that there is this error from the age depth models, which always will be there, I think. And yeah, there is not a silver bullet to this, sadly.
Yeah. All right. Well, that definitely answers my question. It looks like Robert has a question too. Go ahead.
Yeah. Thank you, Ondrej, for this really interesting talk. I found that the data you are requiring are very special, right? So you need these pollen cores. I was thinking, so we are working on a glacier forefield, and there we have a time series as well. So we go, like, we start at the glacier, and then we walk away from it, and thus we have a time series of community development over time. Mm-hmm. And I was thinking our data is relatively similar. So we have the abundances of plant and microbe species. We have a time estimate. We may have also some uncertainty for the time. Do you think your package is like applicable for like general time series on community assembly, or is it really specific to your problem?
100%. I really believe that that you can apply this to your data without a problem, mm-hmm. because you just have to structure it in a way that it will work. But you can see how it should be done. But I try to do it as easy as possible, and also if you do it wrong, the package will give you show. Be careful, I didn't find this and this and this. So it should be relatively user-friendly. But I think even if you don't have the age uncertainties and you have your community data and then your ages, you can still use it. You'll just tell it, "Ooh, I don't have the age uncertainty." Basically, you can turn on and off various aspects of the package, and in the end, the biggest bare bone of what you can do will be just the community matrix, the ages calculated between individual levels, do like a, I don't know, Euclidean distance, right? Without any standardization, because you're using, I don't know, for example, chemical values. It doesn't have to be even community matrix, right? And it will still calculate the rate of change between the subsequent levels, right? This is always the same, and this package is able to do it like without the problem. The more, if you want to do it more intrigued things, like you can do it, okay, we can bin the data, we can include the age uncertainties, we can do some standardization, if you want to use, if you have abundances or if you have data which is possible to do some standardization, then you can start adding these addition, but you don't have to. But I think this is, in my eyes, was the vision of this package, which we will slowly be able to use all various things. Of course, it was originally built just for Poland, but as we were building it, we realized actually we will be able to use all the kinds of different data, and actually many other people will be able to use it. So we'll slowly be adding things to make it more user-friendly and be able to use different data. So I think you should be able to use it without a problem. And I now speak with people who are working with like a coral marine biology, and they're probably going to be using, because that's just abundancy. They're not using abundancy, but they're using like growth, you know, of coral per time, and they will do radiocarbon. So they have H uncertainties of the corals, and they will probably using the package because this is, you know, possible to do. And the data is just very similar. The questions are different, but the methodology is very the same.
Yeah, I will give it a try, and otherwise I will contact.
Exactly. If it doesn't work, contact me, and I will try to improve it so it does work.
Thanks. Great.
We have a comment from Michael Greenacre, who says, fantastic talk. Your data are pure compositional, as in geochemistry and biochemistry. Are you aware of the large literature on compositional data analysis based on the principle of subcompositional coherence? If not, I can write you very relevant here for the standardization of the dissimilarity choice. So are you aware of the subcompositional coherence principle?
I am, to be honest, I'm not. So at least I'm not 100% sure if I know what he's referring to. So if you want to contact me and refer me some, I will be very interested in. That'll be very good. Yeah.
Oh, cool.
So this is, yeah.
Was there any follow-up to that?
I don't know.
Oh, no, I was asking Michael. But
yeah.
okay, so do we have any more questions? Again, I'm watching both the YouTube feed and here and Zoom. So if anybody works with pollen or works with paleo data, I think this would be the guy to ask. All right, well, again, if anybody thinks of questions later on, this post will be in the Ecology and R group, and it will also be in the YouTube channel. So anybody that wants to reach out to Ondrej, then you can contact him on the emails and contact methods that he provided, or message him on his GitHub page and tell him that you're trying to use his package for your data, especially if it's not working.
Exactly. Perfect.
All right. Well, thank you again for giving us some of your time to talk. Our next speaker and our final speaker is Robert Junker from the University of Marburg, Germany. So I'll leave it to you, Robert.
Yeah, thank you, Russell, very much for putting this conference together, and also thank you for everybody who is listening on a Sunday morning to statistics. So that is impressive that there are some interested so much in statistics that you spent your Sunday morning on that. So I will try to share my screen. So can you see now full screen my?
Yep. Looks good.
Perfect. Okay, so I will introduce you to the R package QAD that we recently developed and published. It is all about quantifying asymmetry and direction independence and associations. I will introduce you two papers, and also I will use some R code, and I edit the links to the papers to the chat function. And also the R codes are shared in the chat function at Zoom. So if you want to do it in parallel, or if you want to do it later after the talk, feel free to copy it from there and try the package by yourself. So I will start with what we know about correlations. So in undergraduate statistic courses, we learned you should use a Pearson correlation if you have continuous data, but you should rather use Spearman correlation if we have ordinal data. So, but of course, both of the methods work very well for this example. So here, this is a perfect positive correlation. And of course, the R value and the rho value from Pearson and Spearman, they are 1. So these are perfect positive correlations. The situation is a little bit different here. So you see the association or the relationship between X and Y is not linear anymore. And accordingly, Pearson returns an R value that is lower than 1, but still Spearman detects like the full relationship. It is totally dependent on each other. And so we have a rho of 1. And that reminds us that it's not only about which data we have to do correlation, but also which information we get from these methods. So Pearson is a measure for linearity, whereas Spearman tests whether a relationship is monotonic or not. And that is reflected in the R and rho values here. And as I said, it reminds us that we should think about what we want to learn from a relationship and not only which data we have to use for the methods. The situation is a little bit different here. So this relationship between x1 and x2 is definitely not linear. It's also not monotonic, but we see that there is a strong relationship between x1 and x2. We can use Pearson, of course. It is able to calculate the R value. And you see it's very low because it's not linear. And even if we swap the axis, we will get exactly the same R value. So I exaggerated here with the digits behind the 0 to show you that it is really identical. So Pearson is obviously not able to detect that there is a strong relationship, and it gives exactly the same values for both directions. But when we look at these relationships, we see that the same dependence measure maybe isn't necessarily the right choice, because you can see for each x1 value, we can precisely predict x2 because there is only one option per given x1 value. The situation is a little bit different if x2 is given, because in most of the cases we have 2 options where we can expect x1. So if we know x2, we have, as I said, 2 options for x1. And so the dependence should be a different one. So x2 should be more dependent on x1 because we can precisely predict it, whereas x1 should be less dependent on x2 because we have always these 2 options. And so we have 2 challenges here. First, it is not linear, so we can't use Pearson. Second, it seems to be an asymmetric relationship in the sense that the dependencies of the 2 variables from each other are not the same. And so we could have a solution for that. We could fit a specific nonlinear function to the data. The first problem is we may not always know the function behind the relationship. And so we have trouble in finding the right function. Second, if we have a large data set with many variables and we want to learn about the pairwise dependencies, we will need to spend a long time finding out about these functions. And so that couldn't be the right choice. The second solution could be we should use a dependence measure that finds associations in general situations and quantifies these asymmetries. So let's dig to this solution and see what we can do here. So that is our solution. And for the first part, to find dependence measures in general situations, that can be done, for example, by MIC, the maximum information criterion that was introduced by Reshev 10 years ago. So that method finds dependence in a non-functional situation. So yeah, it just calculates the statistical dependence of two variables. So that is available. However, the second part, and quantifies asymmetries, has not been available for most of the time. And maybe one reason for that goes back to a paper by Renier 60 years ago. And he somehow provided a checklist of properties that should be true for an ideal dependence measure. And I don't go to all the details, but one of his of the properties was it should be symmetric. So we disagree with that property. So we think asymmetry is an inherent property of dependencies, and so we should be able to calculate it. And we lose a lot of information about the dependence if we ignore it. And so we thought we should think about a tool to be able to quantify these asymmetries, and QAD was our result. And so we now can say it does not exist so far, or in other words, now it exists in the form of the R package QAD. When I say we, I say, or I mean Wolfgang Trutschnig, Florian Grissenberger, and myself. So we worked on this method and this R package since some years now. And finally, this year, we were able to publish it in Computational Statistics and Data Analysis. And I guess the audience here is mostly ecologists. And so I also would advise you to check the archive paper that is also linked in the chat function, because here we detailed more about the how you could use it in ecology, how to interpret it. And so there are more practical examples in this paper. But if you refer to the package, please use the publication in Computational Statistics and Data Analysis. The package is available at CRAN, so you can just download the library in R and then use the codes I provided also in the chat function, and I will introduce them later here in the talk. So why would we use QAD? Because other dependence measures than QAD do not work with nonlinear, non-monotonic, and non-functional relationships. So that is true for Pearson, for Spearman, and some other dependence measures. However, there are some that can work on non-functional relationships, for example, MIC, but they usually, or they are symmetric, so they only provide one dependence measure and not two in both directions as we do, and they cannot be used for predictions. And that is also an important function of the R package QAD. So here we provide the option to predict values when we know, let's say, X, we can predict Y, and we propose a different way of predicting variables. I will go into details on that later. So QAD does not require a functional relationship. It has no assumptions on the data, so you don't have to care whether it's normally distributed or whatever. It returns asymmetric or directed dependencies, and it can be used to predict unknown values. So in the following, I will guide you through the R codes that you can use in order to get these dependence measures and the predictions. And in the end, I will show you some examples how we can use it. So first, we use the main function of the package. It's just QAD in brackets X, Y. So these are the two X's. And we save the result as an QAD object here in SR. So I would advise you to do that. It works only also without saving the object, but we will need the object later to further work on the results. So if you do that, you will get this output. And first, you will be reminded what your data are. So x1 is x in our case, x2 is y in our case. We have a sample size of 100. And then for each variable, we get the number of unique ranks. And that number is very important to calculate the resolution. So here for 100 unique ranks, we will have a resolution of 10, which is the square root of the unique ranks. And that has mathematical reasons. You can read them in the paper. But this is well defined, and this is how it works the best way. So, and then we get the dependence measure. And first we get Q x1, x2. And that means how strong x2 is dependent on x1, or in other words, how well we can predict x2 given the knowledge of x1. And here we get the Q value. This is our measure of dependence of 0.814. And that means we can very well predict y or x2 if we have the knowledge of x1 or x in our case here. Then second, we have the dependence measure x2, x1. That means how strong or how well x2 is predicting x1, or how strong x1 is dependent on x2. And we see here the Q value is much lower because for most cases we have two options, and so the dependence can't be as strong as the other way around. Then if you want to use it as a measure of a single dependence for the overall relationship, we give also the maximum dependence. You can extract it from the output here, and then you can use it similarly as an R² value or the MIC value or whatever. So this is the maximum dependence in the whole data set. And then finally, you will get the asymmetry A, and that is the difference between Qx1, x2 and Qx2, x1. And that informs you how asymmetric the dependence is. All of these values are associated with a p-value, informing you about the statistical significance of these dependencies. Yeah, so this is the main function. And as I said, the Q values can be seen as an R² value. The numbers are very similar also in the linear case. So that performs very well. But now we get, as I said, two dependence measures and also this asymmetry that can help you to find interesting relationships in a huge data set or whatever. So next is the plotting function. Now we can plot this QAD object, and we can say, okay, plot it as a copula. And what is a copula? So QAD is based on the theory of copula. A copula is a two-dimensional distribution of data, as you see here. The numbers are bound between 0 and 1, and on both axes we have a uniform distribution of the data. So that is the definition of a copula. And you see here color-coded the density of data in these cells. And based on that, we are calculating the dependence measures. And now you also see why the resolution is important. So here we have a 10 × 10 resolution. And so we calculate the densities in these grid cells in a copula. If you plot it without copula equals true, you see the original data, but also in the same resolution. And here it is rescaled to the original data. So now it has the shape as you plotted it, but still in this aggregated in these cells, and you can see the conditional probabilities for each of these cells. So now let's go to the prediction. Again, you should assign an object, and you use predict QAD with R as QAD object that we defined here. And now predict the Y value for X equals or X1 equals -5, and also show the prediction plot, which is shown here. And with the object $prediction, you will get this output here. And if you use a regression type of prediction or a machine learning algorithm to predict data, you would get a single value as prediction, sometimes associated with a measure of variance or of confidence, but usually it's one value that you can extract, and then this is your prediction. We thought that is sometimes not the best way of reporting predictions, because yeah, sometimes it may be more than one value, or the confidence interval is so large that we should have a better option to look into the data. And so now we get the information how likely X2 is within one of the 10 intervals that we defined by the resolution. And so we get the information with a chance of 40%, X2 is in between 16.8 and 28.7, which is shown here, given that X1 is -5. With a chance of 50%, X2 is in between 28.7 and 38, and with a chance of 10%, it is between 38 and 45. So, and this is now informing very precisely about X2 given the knowledge of X1. And here we can say, okay, a regression type of prediction or a machine learning prediction like in random forest would be totally okay. We would expect something, a value around here, and that would, yeah, give us a good impression. But it's a little bit different if we turn around the prediction and predict X1 given the knowledge of X2. So here we have to say we condition it to X2. We want to predict X1 for X2 equals 70. And of course, we want to see the prediction plot. And this is that case. So here a regression type prediction would predict a value somewhere in between. And random forest would predict either side. So either it would predict the X1 to be here, or it would predict X1 to be here. But none of these answers would be correct, because we see it could be on both sides. And accordingly, our output tells us, okay, there is a 40% chance that X1 is between -10 and -8. And there is a 60% chance for X1 being between 6 and 10, or a 50% chance between 8 and 10. And now we have the full information, and we can use the data to predict certain outcomes with the associated probabilities for each of these events. Yeah, so these are the major functions of our package. And in the following, I will show you some examples how we used it, or how you can use it. For example, this world map here shows the annual mean temperature color-coded. So red means hot and blue means cold. And the annual precipitation is given as the size of the dots. So the larger the dot is, the more precipitation we find in this region of the world. And this is the relationship between annual mean temperature and annual precipitation. And you see we can predict the annual precipitation quite well using the knowledge of the annual mean temperature. But if we have the knowledge on the annual precipitation, we can predict the mean temperature with a lower accuracy. You see this is also non-monotonic, so it's asymmetric. And yeah, we would be better off determining the annual mean temperature instead of precipitation, because now we can predict the annual precipitation much more accurately than the other way around. And this example shows you that if you, for example, have not sufficient time or money or resources to measure a lot of variables in your system, you can use an old data set to identify these variables that are most predictive for the others. And in that case, you can focus on that study, on that variable. Or you can test very precise hypotheses. So if your hypothesis is, I'm interested in how well I can predict the precipitation by the knowledge of the mean temperature, you can say, okay, I can predict it, or the dependence is 0.61. And that is not blurred by the overall statistical dependence between both of these variables. And so now you can formulate directed hypotheses, and you get your numbers to these directed hypotheses. So here we tried different relationships. So in gray in the background, you see what relationship we tested. On the x-axis, you see the noise. So here, what you see here, these relationships, they have little noise, but the further we go to the right, the more noise is included to these functions. And then you see the Q values in both directions, the MIC values, and the Pearson and the Spearman values for these correlations. And in the linear case, all of the dependence measures shown here behave quite similarly. So the more noise we get, the lower is the dependence, obviously. And we see that QAD is a little bit closer to the standard dependence measures for linear relations, Pearson and Spearman, compared to MIC. When we go here to a asymmetric dependence, so here for each X value, we can predict precisely one Y value, but for each Y value, we can predict two X values. We see we have a strong contrast here between QX1, X2 and QX2, X1. So this is why we have these two reddish lines here, whereas MIC and Pearson and Spearman all show only one value, but otherwise they behave differently, similarly. So as you see here, we did this with a number of different examples. So QAD is the only one detecting this direction or this asymmetry, but otherwise it performs equally well as Pearson and Spearman in a linear or monotonic case, and also similar to MIC in these situations. So QAD is well performing. It's comparable to Pearson's R and Spearman's Rho, but it gives you the additional information on the direction and the asymmetry of dependence. So we also tried to use these dependence measures in network inference. And here we simulated data sets that form modules. So the green module, these are variables 1 to variable 4. And you see how they depend on each other. So some dependencies can be monotonic or can be at least measured by Pearson. Others are clearly asymmetric. Then we have here the yellow, the blue, and the pink module, and they are obviously, at least the yellow and the pink one, are obviously not linear, not monotonic. And so Pearson has trouble detecting them. So he can find some of the green relationships, but it completely ignores the pink and the yellow relationship. But that's okay because they are not linear. The same would be true for Spearman, which is more commonly used in network inference. But obviously there is some information missing in these networks. When we use MIC, the maximum information criterion, now you see all of the relationships are found. So we see that this is much better and much better informing about the real dependencies in this system. But if we use QAD to infer the network structure, we get even more information. That time we have two arrows between the variables, so one in each direction. And for example, here the yellow relationship is very asymmetric, and we can predict variable 6 by the knowledge of variable 5 very precisely. So we have a Q value of 0.7, nearly 0.7, but we cannot predict variable 5 by the knowledge of variable 6. Yeah, so that becomes clear here in this sinus curve related relationship. And this information is included in these networks. And now we can calculate motives, we can infer how the relationship is between these variables. So whether it is a mutual dependence, whether it is a directed dependence, and so forth. So now we have much more information than when we use the other methods. Okay, I'm coming to an end, and I will summarize why we should use QAD more often now that it is available. So first, it has no assumptions on the distribution of the data. So you can use it whenever or on whichever data you have. Don't care about any distributions of the data or any other properties. It is freely available as R package with many more features to explore. So you can do a little bit more than I presented here, but yeah, more or less that's the main functions. It is the first applicable directed and asymmetric dependence measure. It's Q, so the Q value is the variable we are interested in. It is possible to make predictions, and here we introduce a different kind of prediction with these probabilities that the predicted value is in between certain intervals. And so that is more, it is capturing all the uncertainty in the prediction. And it also provides you the option to get prediction for 2 or more areas where the predicted value could be. And that is especially important in cases where we have non-monotonic relationships. And because we can now calculate the asymmetry of dependence A, we can calculate directed measures of dependence. That gives a lot of more information than other dependence measures. It may help to carefully, and here I have to pronounce the carefully, causality. So, of course, statistical dependence is not the same as causality. That is something you need to remind. But we did tests and we found that QAD is performing relatively well in predicting the causing variable and the dependent variable in a set of data. And it performs equally well as methods that have been proposed to exactly do that, to infer causality. So it is performing quite well on that, but never stop thinking about it. It is not the same as causality. QAD can also help you to identify key variables or key species in your data. So you can now identify those variables or species in your data set that predict others to a large extent, but that are not that well predicted by the others. And so that seems to be a very important variable in the system, or a species in the system, because it is affecting many others, but is the variation in this species is largely independent on the others. And as I said, you can now test very specific or very precise hypotheses, directed hypotheses. So if you're interested in the effect of variable X on variable Y, you can specifically quantify the dependence in that direction. And you don't have to consider the other direction that may be completely different. So with that, I'm thanking you a lot for your attention on a Sunday morning. If you have questions, I'm happy to answer them. Otherwise, please have a look at our papers, at the R package itself, or contact me for any questions that may arise later when you try the package. Thank you very much.
Thank you for your talk. So we do have a couple of questions and comments, and I have some questions myself as well. We have one person from YouTube, Martin, who asks, so we keep using Spearman or Pearson's. Is there any disadvantages? Do we miss out on dependencies in the past?
Yeah, that depends on what you're looking for. So if you're interested in monotonic or linear relationships, I think Spearman and Pearson are still the best choices. So they are well developed, they are well established, they have a strong statistical power in detecting these relationships. QAD is nearly as good as them in detecting these relationships in a linear and monotonic case. But I think, so there is nothing wrong about using them, that's for sure. But I would still advise to use QAD more often, because now we can get more information out of the same data. So as I said, we get 2 Q values in each direction 1. And that per se is more information than we get for the other methods. And that can be very helpful in interpreting the result, in learning about the relationship between these 2 variables. And it can also help you to identify pairs of special interest in huge data sets. So I wouldn't say it is replacing Spearman or Pearson, but it is a very good alternative, or adding information to these indices. So that is how I would see it. But of course, if you want to get more information, use QAD, and that will help you to understand your data a little bit better than using other methods.
Martin also followed up and said, which value then would we cite instead of one correlation coefficient and p-value?
Yeah, we would cite the QX1, X2 value and the QX2, X1 value with the p-values. We could also give the asymmetry A with a p-value. And I would always recommend to also report the resolution of the data, because if you have a resolution lower than 3, that means you have 9 or 9 to 15 samples. That is working, but be careful in interpreting it. If you have a resolution of 4 or higher, then you have a very robust estimate of the Q values. And so I would always recommend to reporting on the resolution as well, or the sample size or the unique ranks. But anyhow, something that informs about the sample size and the resolution which is resulting from the sample size. Yeah. Or okay, if you want to directly compare it to let's say a MIG, or to have a measure that is comparable to what you get in MIG or what you get in Pearson or Spearman, then you can also give or only give the maximum dependence. And that is a good compromise. If you want to report only one Q value, you should use the maximum dependence. But then again, you're losing the information. That is the good thing about QAD, to have this directed dependence. So go for both, I would recommend.
Okay, and we have a comment from Michael Greenacre. I will try to formulate it as a question. So Wouldn't would this not work well for small to medium-sized samples, since randomization of values within the square root of n by square root of n grid will give the same result? Couldn't that change the data a lot?
I don't get the randomization part because we don't do that except for getting the p-value. But as I said, if you can start working with it if you have 9 samples, because then you have a resolution of 3, but that is really not advisable. If you have 16 or more samples, then I would say it provides robust results. The statisticians I'm working with would say use 10,000 data points, then you are very precise. But of course, that is not realistic. And I would say, so I played around with the package or with the tool since a few years now for all of my data. And from my perspective, with 16 samples, you're on the safe side. 9 or more is okay. If you have 50 or 100 data points, then it's clearly very robust. And I don't get the randomization part, so maybe you can specify that.
Yeah, so he said if you randomize within the blocks of that grid, you change the data, but you get the same Q values. Does that make sense? I suspect he's talking about the blocks of data when you drew out the rectangles of connections on them, yeah?
Yeah. No, I wouldn't agree. So if you randomize the blocks in these plots, then you would have most likely a lower Q value. So let's say you have, let's stick to a linear relationship. So you would have this, this shape of these densities in the copula. And if you would now randomize them, that would look like an independent relationship. So they wouldn't be dependent anymore. And I would say by randomizing the data would most likely always result in lower Q values, whereas the real relationship should have higher Q values. So I'm not sure whether this is fully answering the question, but this is at least what I'm getting from it.
Yep. And I have a question myself too about the functionality of the package itself. You showed single examples, and I ran through the R code with you while you were doing it, and then later on you introduced multiple different shapes. Is there a way to run QAD, the function itself, on multiple, not just XY, but multiple irregular or asymmetric relationships? So using your example of temperature and precipitation, if I wanted to do a monthly check of that over like 10 years, would I be able to do that, or would I need to write a loop function or something?
No, there is the function pairwise.qad, I think it is. Maybe you have a look at the help page, but I think it's pairwise.qad. And then you enter a data frame, and then you will get the like a dependence matrix. It's asymmetric, and you will be informed about the dependence between all the species or the variable pairs, and the lower half will give you the dependence in one direction, and the upper half of the matrix will give you the dependence in the other direction. And then you can use it as a normal dependence matrix or whatever. But remember, it will be asymmetric, so both halves will be relevant. Yeah.
So, and I did have one other question. You mentioned that this is directly kind of competing with the MIC, right? So is there any situation in which MIC would outperform QAD, in which we should select one or the other?
Well, we just submitted a paper where we compared 8 different dependence measures. And so the the power of QAD was a little bit higher than the power for MIC or MIC. So I would say the performance is comparable between both of them. But of course, MIC is a symmetric and not directed dependence measure. And so if you want to get information about the direction or the asymmetry of dependence, then of course you have to choose QAD. If you are just interested in the statistical dependence between two variables or more, then MIC and QAD would provide relatively similar values, at least the maximum dependence, which is the higher Q value of the pair of Q values, and the MIC value would be very well correlated. So as I said, it's depending on what you want to do. If you just want to learn about the statistical dependence, a non-functional statistical dependence, MIC and QAD is exchangeable. If you want to learn about the direction and the asymmetry, then use QAD. And of course, if that is providing more information and gives you more flexibility what you do with the data and what you can learn from the data, of course, I would recommend you QAD. Yeah.
That's a good answer. Okay, so we have one more comment from Michael Greenacre on the blocks. I think he's trying to clarify what he had meant. So regarding the randomization, he means not randomizing the blocks inside the blocks. So with a block, you have the X and Y values. So just randomly disconnect them. So you'll have different data set, but identical Q values. So the square root of N is then critical, and N has to be big. I'm sorry, this is beyond my level of knowledge at this point. So I'm just reading it out and hoping.
Yeah. So maybe I share the screen again. So maybe we can.
Sure.
You now see the PowerPoint again, right?
Yes.
So maybe we can try together to figure out what he means. So do you mean we would shuffle around each of these squares here somewhere?
I will let you know when he, okay, so he says no.
Yeah.
1 second. So based on his, yeah, let me add Michael in so he can. Okay. Are you there?
Hi, Michael.
Are you on, Michael?
Okay, can you hear me now?
Yeah.
Hello.
Okay, listen. So it's much easier talking about this. Is terrible having to write these little messages.
Yeah,
Look,
sorry about that.
In every one of these little blocks, there's a subset of your data, I gather. There's some X, Y, and X and Y values in each block, no?
Yeah, right.
Okay, so now I just randomly disconnect those X and Y values. I just, let's keep the Y's fixed inside a block, and I just randomly reorder the X's. So I do that for every block. So then I will have a different data set, but I'll have exactly the same results because I'll have everything in the blocks again, but the data will be different. That's what I meant. So
you mean within each of these squares?
Yes.
You have a different distribution of the points.
Well, I think there's this different subset of the data inside those blocks, no?
Yeah. So here are a lot of data. So because here it is quite dark.
So just disconnect the X and Y values in that block randomly. Okay.
But they will remain in this square.
They stay in the square, of course. Yes.
Okay.
Yeah. And then you'll have a different data set. If you do that for each block, you'll have a different data set, X and Y values, but all the X and Y values will fall in the same blocks.
Yeah.
So you'll get the same results. You'll get the same results based on a different data set. That's what I was trying to say. So what I'm saying is that the size of those little blocks is very critical, because if they were big blocks, like you only have 9, you said 3 by 3, then you could change the data a huge amount and get the same result. But if you had 1,000 by 1,000, of course the blocks would be small. That's what I was trying to say.
Yeah. So I think if you do these small changes in the structure of the data, it would be also true for Pearson correlation. So if you change them a little bit, all of them randomly, you will get a fairly similar R value.
But you're saying small changes, but that's because you're thinking of the blocks are small. I'm saying if it was a small data set, the blocks would be big, and then the changes would be very big. Okay, that's my whole point.
Yeah, okay, but in that case, you are fully right. So it's all about the resolution.
Yeah, exactly.
But yeah, you're right. So if you
so just as a just as a caveat for our listeners and also myself, who aren't really that statistically inclined as both of you guys, can you explain a little bit how to
yeah, maybe I can summarize what Michael was asking.
Thank you.
So let's assume, so these are 100 data points, yeah? So this is the original data. And the color of each of these blocks informs us about the density of points within each of these squares. And the question was, so let's assume in here are 4 data points. Yeah, and they are, let's say in each corner is 1. And now Michael was asking if we distribute these 4 differently in here. So let's say instead of each corner is 1 point, one is in the middle, the other one is here, here, and here, or whatever. That would change the data, but we would still get exactly the same Q value.
Based on the resolution of the blocks. Okay.
And so now he was asking if we have a low resolution, let's say 3 times 3, we would have larger blocks, so 9 in total. And then we could have a lot of variation in the data within each of these blocks, but always we would get the same Q value. And that is something that is true for this method. And this is why I said, so there will be a warning when you have a resolution of 3 or lower. So there will be warning, be cautious with the interpretation of the data. It is, it may not reflect the full truth of the relationship. But if we have a higher resolution, then I'd say the result is quite robust. And well, we assume that the variation within each of these squares is in any case lower than the variation on the whole axis. And therefore, I think it's a byproduct of the method, but it isn't providing wrong answers because of that, I would say.
All right, that definitely makes a lot more sense. I get what you guys are talking about.
Okay, good.
So the resolution and
thanks, Michael. That is an interesting question.
Yeah, thanks for sure. Okay, so I think now we're going to end it here and let everybody enjoy the rest of their weekend. Sorry for taking everybody's time. Hope you guys enjoyed everything, and thank you very, very much for your talk, Robert. That was a really, really good, a new method for me that I've never heard of, and I will definitely try to incorporate it into some of my research and do some comparisons later on and get back to you on that as well.
Good.
Okay, so
maybe Russell, thank you for putting this together and for organizing it. I know it was it wasn't easy for you all the time, but in the end, it was a great conference. Thank you very much.
Yeah, I
for your
appreciate it.
Yeah.
Okay, so and thank you for every one of the other speakers that came here today. Thank you for the people that registered that are watching on Zoom, and thank you to everybody that is watching currently on YouTube. I appreciate everybody dedicating their time to come out and kind of volunteer to learn and to educate everyone. This is the final day of the Ecology in R web conference series. It went a lot better than I thought it would. I figured we would have a lot more technical difficulties. We had a little bit of trouble in the beginning, but then it turned out to be fine. I learned a lot from watching all of these, and I'm sure everybody else that joined us did. Again, all of these talks are going to be stored on YouTube as well as the Ecology in R page. So if you want to continue the discussion afterwards, if you have more questions for the package developers or about any of the methodology that's used in it, please feel free to start a discussion in the group or even on the YouTube page, but I won't be monitoring that as closely. Everybody here today gave some of their contact information, so I encourage you guys to also reach out to them. Big thanks again, everybody, for making this happen, and I appreciate everybody. Bye.
Russell, you have to do it again, eh?
Yeah, I hope so. I think this went pretty good. I think I'll do it again, maybe in the near future, maybe in a little bit more distant future when I have more time. Oh, and by the way, thank you, Michael, for letting us use your songs. That was like, thank you for introducing me to statistical modeling songs. I didn't even know that that genre of music existed. It might be my new favorite.
I've got a couple more coming. Eh? The data miners song, and I'll build a biplot to paradise.
Nice. Well, feel free to post it in the group. I'm sure everybody that heard them this past week wants to hear them as well.